It's a great pleasure to welcome you at this seminar in the series of the Department of Epidemiology of the Harvard Chan School. And it's my distinct pleasure and honor to introduce Dr. Peter Kraft. Dr. Kraft is the director of the Transdivisional Research Program in the National Cancer Institute's Division of Cancer Epidemiology and Genetics. He focuses on statistical techniques, methods for large-scale observational studies. He will talk about that, genome-wide association studies, amongst others, in cancer. And before joining the NCI, Dr. Kraft was in our Department of Epidemiology. So we know, Pete, we know you very well, and we are very grateful that you are still an adjunct professor in our Department of Epidemiology of the Harvard School of Public Health. It's a great pleasure to ask you to speak about breast cancer, genome-wide association studies, the end of the beginning. Dr. Peter Kraft.
Great. Thank you, Bert. Thanks for the invitation. It's great to see so many familiar and friendly faces and names today. I think I'll be raising at least as many, if not more, questions as answers, and I'd love to hear people's thoughts. Hopefully we'll get into that in the online discussion, but if not, we can catch up later. So I'm going to start to share my screen. And for folks who are watching the recording later, I put my contact info here on this slide. My various email addresses and my socials, so please feel free to reach out there. Okay, so let's get started. The fact that cancer tends to cluster in family has been observed for a long time, if not thousands of years, certainly several hundred. This is a recent modern example from the late 19th century of a cancer pedigree. This is from Paul Broca's book on cancer, where he described the cancer incidence in his wife's family. And in the course of the 20th century, with the development of Mendelian and statistical genetics and eventually genetic epidemiology, we've developed sophisticated ways to quantify this familial aggregation. So one way to do that is to look at familial relative risks. So this is the increased risk to a relative of somebody who has cancer. And you can see here from a classic paper from Neil Risch that these familial relative risks for cancer tend to be around 2, or for testicular cancer as high as 8.5. Breast cancer is right in there at 1.8. We usually quote it as around 2 for back of the envelope calculations. So of course, there are lots of reasons why things might cluster in families. It could be a shared environment as well as a shared inherited genetic component. But there are a number of lines of evidence that suggest, at least for cancer, that this clustering is largely due to genetics. So here are a couple of references from twin registry analyses. Again, a classic paper from Lichtenstein in the New England Journal in 2001. And there was a follow-up study led by Lorelei Mucci in JAMA in 2016. And I wouldn't hang too much on these particular numbers, but the main take-home is that this familial clustering that we're seeing is likely not driven entirely or mainly by shared environmental factors. It's more likely to be driven by inherited genetic, germline genetic factors. Of course, through the first half of the 20th century, although people were fairly convinced they knew sort of in theory how inherited Mendelian characters worked, we didn't actually know the biological mechanism. So it wasn't until Watson and Crick and Rosamund Franklin's discovery of DNA and the central dogma that we started to understand some of those mechanisms, and then we're able to actually measure genetic variation and link that to variation in traits and diseases. So early on, it was expensive to do this. And the number of markers that we could actually measure in the lab were limited. And that meant that folks largely focused on high penetrance families. So families with many affected individuals where they could track the co-segregation or the co-inheritance of disease and particular alleles within a pedigree. And for cancer, that led to a few discoveries, a few high impact discoveries. So this is from one of the initial papers identifying loci that are associated with breast cancer. So this is would eventually become the BRCA1 locus. So this was very important for identifying what turned out to be rare alleles with large effects, relative risks on the order of 20 or more, that were incredibly important for the individuals who carried them and their family members, but didn't actually explain a lot of that familiar clustering that we were seeing, that there were still likely other genetic factors out there that we hadn't identified yet. So in the 2000s, advances in technology and our understanding of the patterns of genomic variation in human populations allowed us to undertake genome-wide association studies. And this so allowed us to measure hundreds of thousands and eventually millions of varying sites across the genome, and then conduct large classical epidemiology studies, cohort studies, case control studies, cross-sectional studies, to identify genetic variants that were associated with traits and diseases. And I'll come to the results of genome-wide association studies in breast cancer in a second. Broadly, genome-wide associations have been fantastically effective in identifying loci that are associated with risk of human traits and complex diseases. And I think the question is, what do we do with these now? And there are sort of two, in my mind anyway, we're walking on two legs. There are two related but disparate goals here. One is to understand basic biology. So if we can understand which loci, what the genes are, what the variants are at these that are driving these associations, how they affect underlying biology, what genes they're affecting, and how that contributes to the development of disease, that'll tell us something more about the biology of disease, which then could lead, for example, to improved therapies. So there are already a couple of examples of this coming out of where the results of genome-wide association studies have led to the development of drugs or therapies. So PCSK9 inhibitors for controlling lipid levels is one. Gene therapies for sickle cell disease is another. So that's basic biology. That is a long game. Identifying a number of loci doesn't automatically mean we understand what they're doing. There's a lot of work that has to come after that. So that will pay off, but it's not necessarily here right now all the time. On the other hand, we might be able to use the variants that we found to stratify people according to risk, and that would allow us to undertake stratified screening programs, for example, or stratified chemoprevention. So to date, genome-wide association studies in breast cancer have identified 239 distinct loci, which account for some, but not all, of the genetic variability that we think is contributing to breast cancer risk. And although the breast cancer genome-wide studies are among the largest that have been done for cancer, there's still more to learn. And like many genome-wide association studies, they've largely been restricted to one or two genetic ancestry groups. So the early genome-wide association studies of breast cancer were almost entirely done in populations of European ancestry, so folks from Europe, or US non-Hispanic whites. Um, uh, uh, Over the course of the last 15 years, more studies in other populations have been done. So the Faherman et al. Was a genome-wide association study of Hispanic and Latino women. There have been studies in African American and East Asian individuals. But again, largely when those are published, it's restricted to that particular group. And there have been a couple of efforts to combine across groups, but it's usually not comprehensive. It's one or two sets of summary statistics where the summary statistics were run by roughly each genetic ancestry group. So that has implications for what we've learned. So there's a couple of limitations from that. The first is we've almost certainly only assayed a subset of genetic variability that's out there in humans. So there are variants that might be relatively common in some populations that are just uncommon in the populations we've happened to sample. We are really underpowered in our current samples to identify those variants. So increasing the diversity in our studies will boost the power to identify genetic variants that are associated with breast cancer or other complex diseases. And the other limitation, as I'll highlight in a second, is that the fact that our data sets are largely of European ancestry and we have smaller numbers in other genetic ancestry groups, it limits the transportability of the results that we're finding. So if we do want to use the results that come out of these studies to stratify people according to risk, those risk models, those genetic risk models often do not perform as well in studies other than folks of European ancestry. So that's an equity problem. We want to make sure that we can develop tools that work well for everybody. So speaking of risk stratification, so just as a quick intro for those who haven't heard of polygenic risk scores, the basic idea is that by combining information across many loci, even if they have small effect individually, we can describe a useful gradient of risk in the target population. So the figure on the right is from a paper that we published 10 years ago now, where we took the loci that were known in 2007, 2009, 2011, et cetera, and then just asked if we could combine the information from those known loci, how much of a risk gradient could we explain. So that first red line there, that's just a step function. That's the risk gradient you could describe back in 2007 when we had one single nucleotide polymorphism that came out of genome-wide association studies. So that's why there are 3 levels here. This is the 3 genotypes. And as you can see, the risk gradient is quite modest. It's very difficult to come up with a scenario where that risk gradient is going to be clinically useful. But as genome-wide association studies got better, and as our methods for developing these polygenic risk scores got fancier, we could describe larger and larger genetic variant, genetic risk gradient, to the point where now we can identify individuals who, out at the tail, say the top 1%, top 2%, who are 3 to 4 times the average risk in the population. And once you start getting out to 3 to 4 times average risk, that starts to be maybe clinically useful, at least in the breast cancer context. If you're thinking of, should I start screening at an earlier age? Should I get screened more often? So then that's just what I'm highlighting here on the left. The curves there are the 10-year absolute risk to an individual depending on their current age. And the red line is sort of a clinically actionable threshold. So above that, you should start screening. Below that, you can wait. And you can see, depending on where you are on the polygenic risk score, the percentiles, there are folks who are at the 99th percentile down to the 1st percentile. Depending on where you fall on that gradient, where you cross that threshold will differ. So that's one potential clinical use of these polygenic scores. And then on the right is work that we did. This is from a PhD student, former PhD student, Chi Gao at Harvard, where we looked at the risk gradient described by polygenic risk scores among carriers of pathogenic variants in high-risk genes. So there, if you think of 20% lifetime risk as an actionable threshold, so that's on the x-axis there is lifetime risk. So if you're above that, then there are a number of professional societies that recommend that you get screening by MRI. So if you're a BRCA1 or BRCA2 carrier, it doesn't matter what your polygenic risk score is, you're above that threshold. On the other hand, if you're a CHEK2 carrier, in the absence of polygenic risk information, your lifetime risk is right around 0.2. You are right on that threshold, and there's a lot of discussion about whether it makes sense for folks who are CHEK2 carriers to get screened by MRI or not. But if you're a CHEK2 carrier and you have a very high PRS, you're going to be well above that 0.2 threshold, and arguably it makes a lot of sense for you to consider MRI screening. So these are just reasons why we might care about these polygenic risk scores. And then just to emphasize the point that I was making earlier, is that the performance of these polygenic risk scores differs depending on the relationship between the target population and the population that was used to train the model. So this is from a landmark paper by Alicia Martin back in 2019, showing that if you trained a polygenic risk score in largely European ancestry population or sample, and then you ask how does it perform in samples from other populations, you see a degradation in performance to where the polygenic risk score is performing almost 4 times less well in African ancestry samples than it was in the original training sample. Recent work has really emphasized that this is, it's not that folks come in different groups, that there's actually a continuum of individuals, and so the performance of the polygenic risk score is really a function of your sort of genetic distance. How similar are you to the population that was used to train the polygenic risk scores? Like, are you represented in that sample? And the further you are away from that sample, the less well the polygenic risk score is going to perform. So that's what's illustrated here. So that y-axis, the x-axis is the distance from the training population. And then the other thing that this is emphasizing is that, people don't come in discrete groups. So you can, classify folks either based on self-report or using a clustering algorithm where you try and map people to be similar to genetic reference samples. But whenever you cluster people like that, there's inevitably going to be some overlap. So you can here see the people who are labeled green, red, blue, or purple, they're all sort of overlapping. Okay, so this is my reminder that that's sort of where we've been. And now let's talk about where we're going in the future. And part of the future for breast cancer, GWAS anyway, is going to be the Confluence project. So just to recap, the familial relative risk for breast cancer can be partly attributed to known genetic variants, whether those are rare high-risk penetrant variants in BRCA1, BRCA2, PALB2, CHEK2, et cetera, or the variants that have come out of genome-wide association studies and can be incorporated in polygenic risk scores. So those are the blue and orange wedges here. There's also a number of lines of evidence that suggest there's more variants out there that are tagged by the SNPs that we can study in genome-wide association studies, but we just haven't nailed down exactly where they are yet. So one of those methods is LD score regression analysis, which was developed at the Harvard Chan School in the PGSG, the Program in Genetic Epi and Statistical Genetics, Alcus Price's group. Another was developed by, that's LDS, the LD score is more of a semi-parametric method, if you will. And there's another parametric method called Genesis, which was developed by Nilanjan Chatterjee's group. Nilanjan's been very involved in the Confluence study. But they both basically say there's stuff we know, there's a lot, we know a lot more is unknown, like there's stuff we know is out there that we haven't found yet. So that raises the opportunity that if we did a bigger study, both a bigger study and a more diverse study, that we should be able to identify additional loci. We'll do a better job fine mapping, which is really pinning down what are the variants that are driving these association signals. We'll be able to improve our polygenic scores, not only by sort of increasing the variance explained, the genetic variants explained by these polygenic scores, but also improving their transportability because our trading samples will be more diverse. And we'll also be able to understand outcomes better. So breast cancer GWAS to date have largely been focused on incidence and maybe by some broad subtypes. What increases your risk of getting diagnosed with breast cancer in general? What increases your risk of getting diagnosed with ER negative or ER positive breast cancer? But where we haven't been as successful is understanding genetic variants that might influence response to treatment and survival. So the Confluence project was launched to conduct the next largest breast cancer GWAS to date in order to address some of those opportunities. And another thing that was important for Confluence, which I'll touch on in a second, is we wanted to create a reusable data resource for the cancer research community. So this is not just going to sit on the hard drive in one institution that a couple of PIs can access, but it really should be a resource that's available broadly. So Confluence is a consortium of consortia. So we're bringing together both existing individual level GWAS data and summary statistics, as well as new genotyping. So the new genotyping I've highlighted here in orange. So the consortia that are involved are the Breast Cancer Association Consortium, SIMBA, which is focused on carriers of BRCA1 and BRCA2 pathogenic variants, Lejano, focused on Hispanic and Latin American women. There are a number of clinical trials which have detailed information on treatment and outcomes. MERGE, which is a study of breast cancer, in the African American Breast Cancer Genetics Consortium. On top of those existing consortia, we've also reached out to a number of emerging biobank studies and legacy studies that are willing to share summary statistics. And we've coordinated here at NCI a number of other studies to participate in the project. So as you can see on the right, the previous largest published GWAS had on the order of 140,000 cases. Now Confluence will have over 400,000 and over 1.2 million controls. And the other thing that's important to emphasize here is that this is a international collaboration. We really are trying to boost the diversity in breast cancer genome-wide association studies. So we're able to have participants from 62 countries involved. So this is a detailed breakdown of the new genotyping that's been performed as part of Confluence, broken down by consortium and genotyping chip. Don't get lost in the details. The key part is there at the bottom in blue. We've been able to really increase the diversity in the samples included. So we have 1.5 times the number of African American samples, and we've tripled the number of Latin American cases. This is, again, sort of breaking down the sample size, but the key thing to emphasize is that most of our data, so this first bit, the new genotyping and existing genotyping data. This is individual level data that we've been able to share, run through a common QC pipeline, run through a common imputation pipeline, and bring together in one analytic space. So we're able to do individual level analyses on 200,000 cases and 171,000 controls. So this is going to be very important for polygenic risk score development, but also for fine mapping, where getting the correlation among markers in your study sample is really important. So over the last 15 years, the field has developed a lot of really cool tools that allow us to take summary statistics and a reference panel to estimate the correlation among markers, to do all kinds of analyses, which is great when for a bunch of logistical reasons, you can't share individual level data and you can still do a lot of the things you want to do. But it turns out it's really difficult to do this kind of fine mapping, which is very sensitive to those correlations in markers in your study. So having the individual level data all in one place is going to be really important and exciting. And at the same time, for locus discovery, we are able to bring in these legacy studies that we're able to share summary statistics to really boost our power to identify new loci. And I'll get back to tease some of those results in a second. But first, this is taken a fair bit of work, not only to coordinate the genotyping and to redo all the QC procedures, get the data in one place, but we've also been very careful about setting up appropriate governance and data management and security and analytic platforms. So in terms of governance, each of the contributing consortia maintains the authority over their data. So they are the ones who are governing their data, but NCI DCEG is serving as a data custodian. So we are setting up the data sharing sites, the data analysis platform in the cloud, that we manage that. But at the end of the day, we're doing so at the behest of the individual level studies, and they're the ones who have to grant access to individual users for particular projects. So on the left there is our Confluence data platform, which you can visit at the URL in the lower right-hand corner. Publicly facing, it has information about the project, the participating consortia, the extensive data dictionary of covariates that we've collected from different studies to enable not just genome-wide association discovery, but also to look at gene-environment interactions, context-specific results, survival. And this is sort of how we're managing requests for data access. So currently, we're still working on our initial analysis, but once we have gone through that and worked out all the kinks in our data, we are making it available to the general research community, and this is where folks can go. They can register on the platform. They can submit a analysis proposal that will then go off to the individual consortia and studies for their review. The nice thing about this platform, for what it's worth, is that this is taking the place. It's managing a lot of this paperwork process for us. So rather than have one person's or multiple people's inboxes fill up with all kinds of data requests and things getting lost, this will allow us to all track that, and it'll all be in one place. And eventually, the approved concepts will be visible to the research community. So that's one aspect of the governance and data management. The other is we are setting up a cloud computing environment for running these analyses, which is scalable and importantly secure. And one of the main reasons we wanted to do this was, well, there's a number of key reasons why we wanted to do this. One was not to just have multiple copies of the data at different stages floating around at different sites. we wanted to have sort of, everybody have access to the latest and greatest and cleanest and version of the data set in one place. And we also didn't want to make this a bottleneck for researchers who might be at a position that doesn't have a high performance computing cluster that would allow them to do some of the analysis they want to do. So we're able to give folks from around the world access to the same set of computing resources. We're able to share code amongst analysts. We're able to share experience. So folks who've never run a GWAS before, we want to be able to give them the opportunity to learn by doing and learn from the other sites who have done this before. So that was one of the reasons why we wanted to set this up in one common environment. And again, for folks who apply for individual level access data, they'll be able to get access to the data through this secure cloud system. Okay. They're just doing all that and being able to do the genome-wide association study with a much larger data set is already incredibly exciting and rewarding. But in the course of doing it, we've sort of had several interesting theoretical problems have come up. And we've had to sort of rethink the way we've done things in the past, partly maybe for what we thought were good reasons, and partly just because that's the way everybody does things. But because we now have these large samples that were all genotyped on the same platform, but are very diverse, we have to revisit the question of how do you conduct a multi-ancestry genome-wide association study? Do you pool everybody, or do you stratify by inferred genetic ancestry group? So this is just a cartoon version of exactly that question. Do we take everybody, run the analysis in the full sample, or do we first break them out into genetically homogeneous subgroups and run the analysis stratified that way? Now, in the past, we haven't had to really struggle with this question. Largely because we have not been able to get individual level data on very highly diverse samples together in one place. So where we were conducting multi-ancestry GWAS, it was usually because we had summary statistics from, say, EPIC, which is done in Europe, and summary statistics from Biobank Japan, which was done in Japan, and then they're automatically stratified. We can't actually get access to the individual data to the pool, so perforce we had to do a meta-analysis. But now we're stuck, we actually could do a pooled analysis. So the question is, which one should we do? So here I'm going to share results from Julie Diaz, who is a PhD student in the biostatistics department at the Chan School. So here we're just illustrating how you might take your initial full pooled sample and break it out into strata. Part of the key point here on the top two panels is that people actually don't come color coded. There aren't pink, brown, beige, green, and blue people. If you were to plot people according to their genetic similarity, and that's what's happening on here and with the principal component. So people who are nearby in those PC plots have on average very similar genotypes. So if you look at that, there's like no clear, well-defined clusters. So what we end up doing in practice is running some clustering algorithms that map people based on their similarity to a set of reference samples, often from the 1,000 Genomes Project. So 1,000 Genomes Project has a number of people who we know exactly where they were sampled from. Often folks will group those into super samples to like, here are all the samples that came from Africa, here are all the samples that came from East Asia, and then use those as reference samples that we then try and say, okay, this study participant, are they most similar to the to the yellow group, or are they most similar to the blue reference population? And then you color code people that way. So that's what's happening in both of these plots. And you can see on the top, which is motivated by or it is from the UK Biobank. If you were to be somewhat stringent and say, I want to restrict to folks who are mapped with high confidence to the yellow, blue, green, beige, and brown clusters, that there are actually a number of folks who fall out of your analysis because they sort of fall in between those cluster groups, or they might get assigned to multiple cluster groups. This breaking people into genetic ancestry groups runs the risk of leaving some folks out, and it also makes it seem like folks come in distinct categories when actually they don't. This genetic similarity is a continuum. And the bottom is from a simulated sample of some very distinct clusters. So these are small groups of people who are very closely related to each other. With some small exceptions, you can see these little gray dots in the middle there, where those would be folks whose parents come from the blue and the green cluster, or from the blue and the yellow cluster. And again, those individuals, if you were to be stringent about assigning folks to genetic ancestry groups, those two folks would fall out of your study. So we want to avoid that if we can. And looking at, so running the analysis pooled might be one way of sort of cutting that Gordian knot. Rather than stress about what's exactly the best way to break people up into groups, maybe we don't break them up into groups at all, and we just run the GWAS in the full sample. So that's what we compared. We looked at the pooled analysis, and we adjusted for principal components that were calculated in the full pooled sample. And we compared that to a meta-analysis where we first broke people into genetic ancestry groups. And then within each genetic ancestry group, we adjusted for the PCs in that substratum, and then did a meta-analysis across the groups. And we were interested in both type 1 error and power. So the major concern with type 1 error is that the pooled analysis may not be robust to population stratification bias. So if the principal components are associated with other environmental exposures, which could happen for all kinds of demographic reasons, or other genetic factors, then the pooled analysis may not adjust for that population stratification bias appropriately. This was one of the arguments why folks were leaning towards the meta-analysis approach in the past. So we were really concerned to make sure that we could control type 1 error. And then we're also interested in power. So I'm mindful of time, so I will just give the bottom line. In terms of type 1 error, we actually do a fairly good job of controlling type 1 error. So on the right panel, panel B, that's when the population stratification is driven by those principal components calculated in the pooled sample. So in that setting where we actually adjust for the principal components in the pooled sample, we're not seeing any inflation, it works like it should. On the left is a situation where the global principal components, the principal components in the pooled sample, may not capture all of the variation that's related to the unmeasured confounders. So if the confounding is better captured by the local ancestry, so principal components, the principal components within each of those strata. In that setting, if there's a large proportion of variance explained, if there's a lot of population stratification going on, then you can start to see some inflation. But as it turns out, in empirical samples, at least for complex traits, including cancer, the level of population stratification is on the order of 0.5%, 0.25% of the total genetic variance being explained by the by the population stratification bias. So that's a setting where pooling still controls for that subtle bias. And in terms of power, the pooled analysis turned out to be the most powerful. We saw that both in simulation studies, and Julie came up with a nice proof of why that has to be the case. And actually, it's more powerful in settings where the causal allele frequency is different across different populations. So the pooled analysis is better even in that setting. So we also looked in real data, and we saw a boost in power there as well. So this is Julie's paper. Do check it out if you want to learn more. There are important caveats, which we can get to maybe in the discussion. But all in all, I think this was very reassuring and led us to answer our question of whether we should pool or stratify with a solid yes. So we'll be doing it both ways, partly because I think we're convinced that pooling will boost our power and still be fairly robust to the kinds of biases we're worried about. At the same time, there will be important analyses where we'll actually want to have the stratified analyses. It's still an open question whether if a variant is only showing up in one of those strata, whether the pooled analysis is actually more powerful. So I'll give some, I will say we are in the middle of doing this right now. We have a draft abstract that we're going to submit to the American Society of Human Genetics, fingers crossed. So in April, we're hoping to have the first public presentation of these results. But as a bit of a sneak peek, I will say that we were able to reliably analyze variants down to 0.05% minor allele frequency. So that's on the order of roughly 100 times smaller than we were able to reliably analyze in earlier studies. And when we wrote up the proposal to justify the budget for doing all the new genotyping, we predicted that we could identify between 187 to 670 new loci. And I'm just going to say we're right on target. It turns out that was a pretty good guess. So I'm just going to mention a couple of open questions, which maybe we'll get to into the discussion, which is the fine mapping is still an open question that we're struggling with. Just as in the initial GWAS discovery, we are not sure whether we should be doing the fine mapping in a pooled sample using everybody, or whether we should stratify by these genetic ancestry groups, because the in-sample correlation structure among variants is so important. And similarly, for doing a lot of the methods that bring in functional annotation to try and understand what are the biological mechanisms through which these variants are acting. Those are also very sensitive to using LD reference panels. So there's a lot of open questions there. We're out of time, which is unfortunate. I really did want to say something about the CONNECT study. So I'm just going to say the CONNECT study exists. So this is a new cohort study being managed out of DC here at DCEG. Target enrollment is 200,000 individuals. We have already enrolled 80,000, on whom we have already GWAS data on about 60,000. We are getting, it's a very sound epidemiologic design. We're getting all kinds of questionnaires and biomarkers from these folks. We're getting early tissue collection from tumor precursors. These are all going to be available to the research community. It's super important that we're going to release these data basically as soon as they come in and we've vetted them, and they're going to be available to the research community. And we are on track for a 2026 release of individual level data. So do watch this space being here. You can Google Connect NCI, it'll come up. All right. I want to acknowledge, some key collaborators for the Confluence work that are presenting. Monse Garcia-Closis was the original DCEG PI on the Confluence project. I've now taken over for her, but she's still very much involved. Tom Ahern is helping coordinate a lot of the platform development and analyses. How you and Julie were key players in the analysis plan and the paper that I was just showing. And for Connect, Mia Gaudet is the senior scientist who is running the cohort, coordinating the cohort, and Nico Wensensen is the PI of record. Of course, both of these projects are the efforts of hundreds of individuals, never mind thousands, or in the case of Confluence, millions of participants who have generously shared their information and data. I will say for Confluence, one of the great pleasures has been working with the Participant Advisory Board. We've learned a lot from them in terms of framing our scientific questions and also in terms of thinking about returning results back to study participants. So with that, I'm going to stop sharing and take some quick questions.
Dr. Kraft, thank you very, very much for this exciting overview. Really very impressive work in Confluence as a consortium of consortia, an Uber consortium, we could say, actually. And also in the Connect study, the Connect cohort to see, really very impressive. Are there questions? I invite them. Are there questions or comments from the audience, please? Dr. Mingyang Song. Dr. Song, please go ahead.
Thank you, Bert. Pete, that was really a phenomenal talk. Thank you so much. I guess I have a quick question. So for the Connect, I wonder if you can comment on like how it differs or like integrates with All of Us study.
Yeah. So, one of the main differences with All of Us is the sampling design. So Connect is based in these healthcare providing organizations. So we are sending out invitations to everybody in those organizations. So everybody who gets care in Kaiser, Colorado, everybody who gets care at Kaiser, Hawaii, everybody Baylor Scott and White in Texas, everybody in the system gets an invitation. So we sort of, in principle, we know what the sampling base is. Whereas if you've followed up on All of Us, there's open enrollment. People can just volunteer. So it's not clear necessarily what the sampling frame is. So that's one of the key reasons. And the other is that it's really focused on cancer. So we're putting a lot of our efforts into collecting exposure information that's relevant for cancer, to linking to cancer registries, so we get good phenotyping. So and also building out mechanisms to get to some of those precursor lesions. So if somebody goes in and gets a biopsy, we know who they are. We can reach out to the hospital and get the biopsy sample and put it in our biobank. So I think those are some of the things that make it different than All of Us. I mean, that said, we are linking to EHR data. So if folks are interested in cardiovascular disease, we will also have EHR phenotypes on some of those on cardiovascular disease, for example.
Okay, thank you.
Any further questions?
And then I noted
there's a comment from Walt Willett in a note, namely All of Us has not collected data on diet, and follow-up seems to be very low.
Right. Yes, so we are okay. Now I should have done my homework before getting on. We are definitely sending out a dietary questionnaire. Just whether we've sent it out is the question. But I know we've done like a physical activity questionnaire. So again, I think a lot of all of us information is coming at baseline from a questionnaire or the EHR, whereas here we're going to have modules that are capturing more detailed information, and we will get repeated measures over time. I will also throw out, one of the longest questionnaires in the baseline, one of the longest sections in the baseline questionnaire was residential history. People are happy to tell us that it took them a long time to fill this out. But they filled it out, and we actually get very detailed information about people's lifetime residential history, which is going to allow us to do some of the GIS mapping to early life exposures for environmental exposure. So we're excited about that.
Thank you. David Hunter has a question, I think. David.
Hey, thanks a lot for the talk. Can you hear me?
Yes.
Fantastic. So usually when we talk about, or people want to criticize polygenic risk scores, they complain, well, all the information is on European ancestry, and that's contributing to disparities. Have you had a chance to look? Are you finding ancestry-specific variants or cosmopolitan variants with radically different relative risks? And can you just comment on the transportability of the breast cancer PRS to other ethnicities?
Yeah, no, it's a good question. So, and I will make 3 points, I think. So, of course, now I've forgotten what all 3 were. Well, in terms of the transportability, there is definitely a loss in the risk gradient explains. If you're thinking of like what's the area under the curve from 313 SNP PRS developed in largely European ancestry population, and you look in a Korean sample or a Malaysian sample or an African American sample, that AUC does come down. There are a number of multi-ancestry methods that allow us to increase the AUC across populations, actually. I think the limitation for those methods to date have just been still the limited training sample sizes outside of European ancestry folks. The bulk of the training data still comes from European ancestry. So I think we'll be able to do much better. In terms of whether alleles actually have difference effects. So I didn't go into it, but one of the main reasons that you see this degradation in performance is due to differences in linkage disequilibrium. So that the SNPs that end up in the polygenic score or the weights that they get is really driven by the other variants that the variants in the score are correlated with. And those patterns of correlation differ from population to population. So something that's a really good proxy in one population may be an proxy in another, or just not a good proxy at all in another population. So that's maybe one of the main reasons why we see this degradation in performance. The other might be like actually at the causal variant, the effect differs. On that question, I still think we don't have like really strong evidence that that's happening to a large degree. I didn't get a chance to throw in the slides, and I meant to. We have a preprint. Hao-Yu is one of the main authors on that, where we looked at the genetic correlation and effects across ancestries, and this is my gloss on it was that there wasn't strong evidence for big differences. But I would take that with a big grain of salt because we still have limited data in a lot of groups. So that just means there's a lot of uncertainty around that conclusion. So, that long story short, the degradation in performance could be happening even though the actual causal variants are present in every population and have the same effect. And the last thing I'll note is, just because you see this degradation in the gradient explained doesn't mean that these polygenic scores are not clinically useful in other populations. So it may be that even though the per standard deviation relative risk goes from 1.7 down to 1.55, that 1.55 gradient of risk is still clinically useful and actionable. So I think we have to be careful not to throw the baby out with the bathwater, as it were. So it is important to note that there may be limitations in the transportability, and we might be able to do better, or we will be able to do better when we have more diverse training samples. That said, it doesn't mean that these things are not useful now across a wide range of settings.
Thank you very much. Can I perhaps finally ask a naive and non-cancer epidemiologist question? Is the heterogeneity of cases, breast cancer cases, is that something of a concern here, men and women combined or whatever you have? Is that something that is important here?
Thank you. It's a great question. It's very important, and definitely something that we have in mind. Again, I guess that's when we talk about diversity, there's not only in terms of the diversity in the genetic ancestry of our participants, but also in terms of outcomes and the types of cancers that we're including. So as it turns out for breast cancer, a lot of the variants are shared across subtypes. So they contribute both to risk of ER positive and ER negative disease. They're associated with risk in the general population and as well as among carriers of BRCA1 or BRCA2. Male, female, similar risks, similar loci.
Yeah.
Often, but not always. There are some variants that are subtype specific, so really associated with either ER negative or even triple negative breast cancer. And it's an open question, honestly, about whether there are variants that are specific to male breast cancer. We are going to have the largest GWAS of male breast cancer, and clearly we'll analyze those separately from the female cancers. That said, numbers are still pretty small.
Thank you. Dr. Kraft, thank you very, very much. This is it's a great seminar. I tremendously enjoyed it, and I think I'm speaking for everybody here in the audience. I wish you all the best, of course, with the government shutdown and other challenges that we share actually in some senses. And I would like to say one final word, namely invite you all to the next seminar, which will be next week, which will be also on Zoom, and it is a joint seminar from Epidemiology of Harvard School of Public Health and the Dana-Farber Cancer Institute. And you will then hear about, again, cancer, but now from the American Cancer Society. Thank you very, very much. Again, Pete, wonderful, and hopefully see you soon in person. Thank you.